I hope you all are doing well. Welcome back to my YouTube channel. In the last video, we did a video that was on cellular life. Okay. If you haven't checked that video out, please go and check that out as well. I have explained the properties and the structure of a virus. And also, I have explained HIV and AIDS. Okay. That is a part of your PMDC syllabus. Right. Now, today the topic which we are going to discuss is the bioenergetics. Okay. So I have made a presentation on bioenergetics, which we are gonna see and discuss. Okay, so what is bioenergetics and what is included in your MDCAT syllabus? We have done topic number 1 in my previous video. If you haven't seen it, I'm gonna write down the link in the description box. You guys can go and check that out. Next, we are moving on to bioenergetics and we will discuss the topic of respiration. Respiration of proteins and fats and correlation with these respiration mechanisms with that of glucose, right? So we are going to discuss the main outline of respiration. Then we are going to discuss aerobic and anaerobic respiration. In addition to that, we will discuss glycolysis, Krebs cycle, okay, gluconeogenesis, all those stuff we are going to see. Okay, so what is bioenergetics? It means the flow of energy, transformation of energy within living systems. Okay, why do organisms need energy? In order to grow, reproduce, survive, right? And they obtain nutrients from food. So now when we take that food in, we took a carbohydrate. For example, I ate bread. Now, what mechanism will it undergo that it releases energy in the form of ATP? This is the mechanism of bioenergetics. Bio means life and energetics. How does why our life needs energy and how does other organisms, living organisms, help to produce energy when we consume them? Okay, right? They perform biological work. Humans or any other animals, they perform biological work. For example, muscular contraction, active transport, biosynthesis. For all of these things, we need adenosine triphosphate. We need energy carrier molecules like NADH, FADH. Wait, this is F. FADH, NADH, okay, ATP. We need all of these, right? So these are produced via bioenergetics, right? Now move on to our topic, which is respiration. You don't have to do bioenergetics in plants. You don't have to do photosynthesis or cytochrome structures, right? We'll just directly move on to the process of cellular respiration. Now, what is cellular respiration? In cells break down organic molecules, mainly glucose, in order to form ATP. So it basically goes like this: glucose plus oxygen produces what? It produces carbon dioxide plus water plus energy in the form of ATP. Right? This is a very simple equation which we have studied since our metric or intermediate, and you must know this by now, right? This is just a quick revision of the topic. Okay, so now we are moving on to two different kinds of respiration, which are aerobic and anaerobic respiration. First, I'm going to give you a brief overview of all of these things so that it builds a foundation for you, right? After that, in the next part, we are going to do complex cycles as well. Okay, now what do you mean by aerobic? It's clear from the name, aerobes means the ones who live with oxygen, okay, the ones who require with oxygen, right? They have to break down glucose with oxygen. It undergoes complete oxidation. Why? Because an abundant supply of oxygen is present, right? There is no supply of oxygen. And occurs in cytoplasm and mitochondria. It requires oxygen. End products are carbon dioxide, water, and ATP, right? Anaerobic respiration, the breakdown of glucose is without oxygen. That is why it is known as partial oxidation. It is not completely oxidized. It occurs only inside the cytoplasm. It does not require oxygen. These are very basic. And anaerobic respiration, it produces lactic acid and ATP. Lactic acid is produced where? It is produced in muscle cells, right? And also another product is ethanol, right? Ethanol is produced where? Ethanol is produced in yeast when we leave it to make bread. So it produces ethanol, carbon dioxide that enables bread to rise, enables the dough to rise. Okay. All right. Now we move on to the ATP yield. Aerobic obviously, when we are giving it lots of oxygen, so it yields more, right? It produces lots of ATP, which are 30 to 32 ATP per molecule of glucose. And anaerobic respiration, it produces low ATP, which are 2 ATP per glucose. So you see that the number of ATPs, number of energy produced is very less. Efficiency, when we talk about that, it is highly efficient. This is inefficient. Why is it inefficient? Because, see, lactic acid is produced in the human body as a result of anaerobic respiration in the mouth, right? Now, in order to cover this lactic acid, we need oxygen again. So it produces a thing that is known as the oxygen depth, right? And this needs to cover. That is why a human being remains panting and remains very sweaty, remains like, breathing very heavily over a long period of time. Why breathing heavily? Because, yeah, why breathing heavily? Because he needs more oxygen in order to fill the oxygen debt. Okay. The byproducts that are produced are carbon dioxide and water, in aerobic respiration, and in anaerobic respiration, it is ethanol and carbon dioxide for yeast, right? And the example is normal metabolism of aerobic respiration, whereas anaerobic respiration, we have yeast fermentation. It occurs in RBCs or in muscle cells during oxygen debt, right? All right, now we are done with the differences between aerobic and anaerobic respiration, and I want you to pause this video and recall all the differences in your mind and the products which are formed. Okay, so take a minute, just pause this video and just recall everything. And if anything is not clear, just remember I am here. Write it down in the comment section, and your queries will be solved, inshallah. All right, next we move on to the anaerobic respiration. First, I'm focusing on anaerobic respiration. Why? Because this is a small topic. Aerobic respiration is a vast topic, very vast topic. So we need to cover lots of things. That is why first let's cover anaerobic respiration. Now we are done with the introduction. Let's discuss the types. The first type is alcoholic fermentation. The second type is the lactic acid fermentation. Okay. Now what is the equation for both of these? The substrate which is used is pyruvate. Okay, the reactant or the substrate is pyruvate in both of them. This is a similarity. While product in alcoholic fermentation, what do you understand by the word alcohol? Alcohol means ethanol. Okay, ethanol is an alcohol, right? So ethanol is produced in addition to this carbon dioxide is also produced, right? And over here in lactic acid fermentation, pyruvate is only converted into lactic acid. Where does alcoholic fermentation occur? This is very easy. It occurs in yeast because it's used to make pizza and breads, and it also occurs in some of the bacteria. Bacteria, for example, those that live in hot springs where oxygen is not available, it is sparse in supply. So those bacteria, which are especially those which are thermophilic bacteria, these are usually undergoing an alcoholic fermentation. Okay, and the site for the lactic acid fermentation is muscle cells, which produces oxygen depth, as we discussed before, and some bacteria. The end products, it's very easy: ethanol, carbon dioxide, and 2 ATP. Whereas over here in lactic acid fermentation, we don't produce carbon dioxide, just lactic acid is produced and 2 ATP are produced as well. So the energy yield for both of them, for alcoholic and for lactic acid fermentation, it is the same. Okay, it does not decrease. Right? Carbon dioxide production does not occur in lactic acid fermentation. You need to remember this when if they give you an equation in the MDCAT MCQ paper that whether fermentation occurs or not, whether carbon dioxide production occurs or not. So you're gonna write no. Okay. Energy yield is the same. Uses are used in baking. Alcohol fermentation is used in baking. But lactic acid fermentation is used for temporary energy in muscle, yogurt, cheese. Okay. And alcohol fermentation, you need to remember this, that whenever the it is given to you, an equation is given to you that whether the arrow is made like this or the arrow is made like this. Okay, this is the arrow of an irreversible reaction, and this is the arrow of a reversible reaction. So alcoholic fermentation is irreversible. When it occurs, it cannot be converted back. Whereas lactic acid fermentation, it is reversible in human beings, right? Whenever muscle cells, they produce lactic acid, this lactic acid can be converted back into pyruvate. And this is known as the Cori cycle. Okay, so when they ask you what is the reversal of lactic acid fermentation in human beings, you're gonna say that it is the Cori cycle. Why the Cori cycle? Because the product of anaerobic fermentation of lactic acid fermentation, which is lactic acid, it can be converted back into the raw material, into the reactant, which is pyruvate. Okay, so just remember this point. Now, this is a diagram from the federal. I have added this in my presentation because I thought that it's very self-explanatory. See, first we have glucose. Glucose undergoes glycolysis and it is converted into pyruvate. We are discuss this thing right now. Okay, don't worry if you don't know about glycolysis or if you don't know how glucose is converted to pyruvate. Okay, we are gonna see all of this. Okay, just don't worry. Wait a second. Okay, so after pyruvate is formed, what happens is that this pyruvate, it basically what it does is that it is converted into lactate. And how is it converted into lactate? This is the first equation in which we have glucose and NAD. See, this NAD will be converted into NADH and 2 hydrogen positive. Okay. So this is the reactant, which is NAD, and it is converted into NADH plus 2H positive. Okay, and this is the pyruvate. Okay, this is pyruvate. Now what happens in the second phase of the reaction? This was the first phase of the reaction. Now in the second phase of the reaction, what happens? This is number 2. What happens over here? Pyruvate reacts with the NADH, which was formed in the above reaction, plus 2 hydrogen in order to form lactate, 2 molecules of lactate and 2 molecules of NAD. Okay, so this NAD H is again converted into NAD positive. All right, first it was produced and now it is converted. And you need to remember the balancing of this situation, that 1 molecule of glucose will produce 2 molecules of pyruvate. Okay, and 2 molecules of NADH and 2 H positive, right? And 2 molecules of pyruvate will produce 2 lactate. It will not produce 1 lactate. 2 molecules of pyruvate produce 2 lactate. 1 molecule of glucose will produce 2 pyruvate. Okay, so you need to remember all of this, right? And in this reaction, by the help of glycolysis, what is the energy yield? It is 2 ATP. We are going to discuss all of this step by step in the next lecture or in this lecture if I have time. So don't worry about this first part. We will discuss this. Just worry about the second part, that is the conversion of pyruvate into lactate with the help of NADH that is being converted into NAD positive. Okay. Next, we move on to the alcoholic fermentation. Now, in alcoholic fermentation, you see that the first step is the same, that glucose is being converted into 2 molecules of pyruvate, right? And when these 2 molecules of pyruvate are here, it is not converted into lactate. Okay, it is converted into what? It is converted into aldehyde, into an aldehyde group, which regenerates NAD and it produces 2-ethanol. So in order to summarize the reaction, we have skipped this step. This is basically the intermediate step, right? Which they have skipped. They have just written this step, this number 1, where glucose is converted into pyruvate, right? And this is the same as the step which is given in the previous one. Yep. See, it's the same. Reaction number 1 is the same in both alcohol and lactic acid fermentation, but reaction number 2 is different. Why? Because here pyruvate is converted with the help of NADH and OH positive into ethanol, not lactic acid. Okay, ethanol, carbon dioxide, and 2-NAD. And this is a reversible reaction. They have shown the intermediate step, but you can see and you can visualize it in the diagram that 1 molecule of glucose converts to pyruvate. 2 pyruvate converts into 2 acetaldehyde, 2 acetaldehyde convert into 2 ethanol, and they via the help of glycolysis 2 ATP are produced, and via alcoholic fermentation what happens is that NADH are used and they are converted into NAD positive. Okay, so just remember all of these facts, and if you have any questions, let me know. All right, now we start the process of aerobic respiration. What is aerobic respiration? We have already discussed it, that aerobes mean the one that use oxygen, and it produces a very high yield and a high energy yield, that is 30 to 32 ATPs. Okay. All right, now we move on to glycolysis. What is glycolysis? Basically, it is a process when one molecule of glucose breaks down into a molecule of pyruvate with the net production of 2 ATP and 2 NADH. See, over here, as we saw, the first reaction, this first reaction is of glycolysis. Okay, the first reaction of glycolysis when one molecule of glucose breaks down into pyruvate and 2 NADH. Okay, and it occurs where, this is a very important point, they can give you like four options: smooth endoplasmic reticulum, mitochondria, cell membrane, or the cytoplasm. So you have to remember that glycolysis occurs in the cytoplasm of all the living cells. It is an anaerobic process, and it is the first step of cellular respiration. Why the first step? Because see, what happens is that in our body, for example, I took bread. Okay, I ate bread. Now what will happen in my body is that digestion will happen, and the end product of digestion, the end sugar which is produced, is glucose. The basic sugar, basic molecule is glucose. Now how this glucose will provide me energy, how this glucose will be converted into other intermediates in order to provide ATP. So the first step from glucose and its conversion into pyruvate and the production of 2 ATP and 2 NADH, this process is known as glycolysis. And that is why this is the first step of cellular respiration, because glucose is the first product that is being converted. Okay. And this is an aerobic process. Okay, you have to remember this, that this does not require oxygen. Oxygen will not participate in glycolysis. Okay, I hope this is clear. If you have any questions, let me know. Now we are going to discuss the diagram. Okay. Wait a second. Okay. You may forward this video a bit, because I'm just gonna fix my screen in such a way that I can read the theory as well, and teach you side by side. Okay. Now we are over here. Here's your reaction, and here is your theory. Okay. All right. So now the first phase is used is the energy investment phase. Now, what do you mean energy investment phase? It means that we are, wait a second, just, yes. What do you mean by energy investment phase? It means that it uses energy. It uses ATP in order to produce intermediate products, right? Now, the first step, as you see in this diagram over here, the first step I have over here is glucose. It is the first intermediate, right? It is converted into glucose phosphate. How is it converted into glucose phosphate? Because my ATP is being converted into ATP. So when ATP is converted into ADP, what happens is one phosphate inorganic molecule. So this phosphate inorganic molecule will go and attach with glucose, and it forms what? Forms glucose 6 phosphate. Okay. Now this glucose 6 phosphate, it rearranges into fructose 6 phosphate. As you can see over here, glucose 6 phosphate, it would rearrange into what? Fructose 6 phosphate. How does it rearrange? Why? Because glucose and fructose both are what? Isomers. Why are they isomers? Because both of them have the same chemical formula, but they have got different structure. Okay, so they can easily rearrange into one or more structures, right? That is why glucose 6 phosphate converted into fructose 6 phosphate. After that, in step number 3, what occurs is second phosphorylation. First phosphorylation occurs with the help of glucose, and second phosphorylation occurs with the help of the fructose. Right now, over here, fructose is being converted into fructose 1,6 bisphosphate. What do you mean by bis? Bis means it means that two phosphates are attached over here. One at position 1 of the carbon, one at position number 6 of the carbon. If I can show you another diagram. Oh no, the structure is not over here. Okay. So see, glucose or fructose, both of them have got 6 carbons, right? If I write over here, 1, 2, 3, 4, 5, 6, right? Now one phosphate will attach over here, P, and the other phosphate will attach over here. One at carbon number 1 and one at carbon number 6. Okay? Right? So first it was only at carbon number 6. First in glucose it was attached at carbon number 6. Now it is attached to carbon number 1 as well as carbon number 6. That is why it is known as fructose 1,6 bisphosphate, and it uses 1 ATP for the reaction. As you can see in this diagram, that 1 ATP is converted into ADP, and second phosphorylation occurs where fructose 6 phosphate is converted into fructose 1,6 bisphosphate. Okay? Right now, why is this known as energy investment phase? Because two ATP are used, one over here and one over here. Okay. Now what happens over here? Now there is cleavage. Okay. What happens in cleavage is that fructose 1,6 bisphosphate, it splits into 3 carbon sugars, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this fructose 1,6 bisphosphate, which previously contained 6 carbons, it split down into 3 carbon structures, both of them which are isomers of each other. ठीक है? These split into DHAP, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate, which are reversible reactions and they can easily convert into one another. Right? After that, isomerization occurs where dihydroxyacetone phosphate, it is converted into another G3P. So it produces 2 G3P. First, it was 1 DHAP plus G3P, right? Now it converted this DHAP converted into G3P and it produces 2 G3P. Okay, 2 molecules of G3P, they are produced from 1 molecule of glucose. All right. As you can see over here, this is a preparatory or the energy investment phase. Let us revise all of this. Okay, before starting the energy payoff phase. First, we have the product of metabolism, that is glucose. Glucose, it uses 1 ATP, converted into ADP, and it is converted into glucose 6-phosphate. Now this glucose 6-phosphate is isomerized and it produces fructose 6-phosphate. Now this fructose 6-phosphate, it again is taken 1 ATP and it is converted into ADP and it produces fructose 1,6 bisphosphate. Okay, now this fructose 1,6 bisphosphate, what happens is that it breaks down, there's a cleavage, it produces glyceraldehyde 3-phosphate G3P and it produces dihydroxyacetone phosphate DHAP, which are interconverted and they convert into one another. This DHAP is converted into G3P. So this was the preparatory or the energy investment phase of glycolysis. I hope this is clear. Now we are starting the energy payoff phase. Okay. Okay. Now we are starting the energy payoff phase that produces 4 ATP and 2 NADH. Okay. Now, if you guys have any question, let me know and ask me. Okay. What happens is that oxidation occurs and NADH is formed. Each G3P is oxidized, it produces 1 NADH per molecule. See, this is G3P which is being oxidized and is producing NADH, right? After that, it produces 1,3 bisphosphoglycerate. Okay? After producing energy, what does it produce? This G3P is converted into bisphosphoglycerate, 1,3 bisphosphoglycerate. Why? Because it has got 2 phosphates which are attached to it, right? See, over here, if I zoom in, wait a second. If I zoom in this, look over here, this is PI. Can you see this thing? PI, PI. What is this? Wait, let me just erase it. I just did something. Okay, now it's erased. See, this is PI. What is this PI? This is phosphate inorganic, right? Which is attaching to what? That is attaching to the G3P. I just lost my cursor. Okay. So it is being converted into PG. Why? Because it adds one phosphate. And in the process, it produces one NADH. After that, what happens is that first ATP formation occurs from what? It occurs from BPG. After BPG, what happens? It produces one ATP and 3-phosphoglycerate is formed, 3PG. Okay, so basically this 1,3 phosphoglycerate, it is converted into 3 phosphoglycerate. Why? Because it gives in one phosphate inorganic where to the ADP. It gives one phosphate to the ADP in order to produce ATP, right? That is why what happens is that ATP formation occurs in both of the isomers. There are basically two going on now, right? After DHAP was converted into G3P, we start reaction twice. Every reaction is starting twice, okay? So what happens over here is that if ATP is produced over here, it is also produced on this side, and by this mechanism, see that two ATP are formed over here, right? Okay. All right. All right, now what happens over here? 3-phosphoglycerate, it is producing 2-phosphoglycerate. Why? Because it occurs, because what happens is that rearrangement occurs, and in that rearrangement, this phosphate, for example, these are 3 carbons of the 3-BPG, and this phosphate shifts from this position at carbon number 3 to the position on carbon number 2. It comes in the middle. Okay, so why is it shifting position? So that it can undergo different chemical reactions. Okay, after 2 2PG, it gives off water, right? And it produces PEP, phosphoenol pyruvate. Okay, you don't need to go in the depth of the reaction that how does this occur and all the chemical processes. You don't need to read that. You just need to understand that 2PG is converted into phosphoenol pyruvate, which is converted into pyruvate, and it produces ATP. Okay, and this is producing ATP. What happens is that one ATP is produced on this side and the other ATP is produced on this side. So the total ATP which are produced are 2 ATP. Okay. And what is final product? The final molecule is pyruvate. It is basically, as you see over here, the final product is 2 molecules of pyruvate. Okay, now let's have a look at the phase. We actually did it from here, but we are gonna, revise it a bit. So over here at glyceride phosphate. What happened in the energy preparatory phase? The last step of the energy preparatory phase was the conversion of DHAP into what? Into GTP. Okay, when it converts into G3P, what happens is that 2 G3P are produced, which produces what? That produces 2 NADH. This is the energy yield. Okay, so you have to learn that at which stage of the reaction what is being produced. Which state of the reaction produces ATP, which stage of the reaction uses ATP. Okay, what is the net yield of the reaction? Then 1,3 phosphoglycerate is being produced. Why? Because one phosphate inorganic is added at this step, right? And after that, this phosphate is removed from in this molecule and it is added to ADP in order to produce ATP. Okay, 2 ATP are produced over here in this stage. And when it loses one phosphate is gone, it only has phosphate on position number 3 now. It becomes 3-phosphoglycerate. Okay. After that, what happens? Rearrangement occurs. It becomes 2-phosphoglycerate, right? And these are the number in the brackets. These are the molecules. Okay. After that, what happens? It gives out water and it becomes PEP, phosphoenol pyruvate, and then it gives out 2 ATP in order to become pyruvate. Why? This phosphoenol pyruvate. It gives a enol group. It gives the phosphate as well. Phosphate. There are two phosphates. Why? Because there are two molecules of PEP. Two molecules of PEP. It means that there are two phosphates over here, right? So it gives one phosphate to one ATP and other phosphate to other ATP. So two ATP are converted into two ATP with the help of this reaction. I hope you guys understand it now. So this is just the same diagram as we just discussed. Now, what is the net gain from glucose? As you see over here, let me just, yeah. Okay. So it's lagging. My PC is lagging. My PC is lagging a lot. I think I need to end the meeting because I don't have space. Okay, anyways, this is just the preparatory phase where, see, over here in step number 1, we use one ATP which is converted into ADP, and step number 3, ATP is converted into ADP, right? In step number 6, we produce NADH, right? Step number 7, ATP is produced, and also in the last step where PEP is converted into pyruvate, there also ATP is produced, okay? Right. Now the net gain from glycolysis. The ATP which I used are 2, produced are 4. So 4 minus 2 makes the net yield, which is 2. NADH are 2 and pyruvate also, 2 pyruvates are produced. Okay, so ending the video here because my laptop is not supporting it. There's lots of information that we need to cover still, right? It's remaining. So in the next video, we are gonna cover the Krebs cycle. Then I have the oxidative decarboxylation, ETC. And lastly, we have what? We have sites of substrate level phosphorylation, cellular respiration, and fats and proteins. So many topics are remaining, and these will be covered in part 2. Okay, and this will be uploaded on YouTube. And if you have any queries, let me know. And if you need the slides, I'm going to attach the number below. Please contact the number so that I can send you these slides. Okay. And if you haven't watched the previous video, let me know. And please watch that video as well. It has been uploaded on my channel about acellular life. So I'm ending the meeting now. All right. Take care. Allah Hafiz.